We will reduce coffee break for 5 minutes, so we have a fair time for María José Alonso. She's a professor at the University of Santiago de Compostela. She's been working on drug delivery for a long time already, from the very beginning, I guess. She's been among the top 10 in pharmacology. She's a good pharmacologist for sure. And also she's been coordinating projects of European projects from FP6, FP4, FP5, FP6, 7, and 2020. And she's currently the secretary of the Controlled Release Society. So for drug delivery with polymeric nanoparticles and steady release of active principles and all the rest, I think María José Alonso is one of the world leaders. So please.
Thank you, Victor, for the kind introduction, and thank you for inviting me to come here and present my work, the work that we are doing at the University of Santiago de Compostela in Spain, just the northwest corner, 1,500 kilometers away from here. I want to thank, obviously, the organizers and the scientific committee for having the idea of inviting me to come here. So basically, my background is pharmaceutics, pharmaceutical technology. So what I would like to tell you today is about the formulation of biologicals, biological drugs, using nanotechnology approaches. When I say biological drugs, what I mean is mainly macromolecules, you know, that the new drugs coming out into the market, most of them are now large molecules like proteins, polynucleotides, monoclonal antibodies, large peptides, and these molecules are very difficult to deliver. So we started working on the development of new formulations for these macromolecules already more than 20 years ago, because we were conscious of the special needs for these drugs to overcome biological barriers and reach their targets. So when I say formulation, what I'm meaning is to give these drugs with the adequate form to be able to accomplish their goal, their effect, once administered into the body. So this is what it means for me, formulation. And the work I'm going to present here, I'm going to present some data which have not been published yet, and the authors of this non-published data are those indicated here, my PhD students, Carmen, Anna New, and the postdoc Mathilde Duran. So let's go. So in our lab, our main principle of work is rational design. We rationally designed nanocarriers for delivering molecules, and in this case today I'm going to tell you for delivering macromolecules, because this is our specialty. So when I say rational design, what do I mean by that? What I mean is that based on the current knowledge we have about the biology, about the barriers, you know, the biological barriers, and about the drug properties, we design these nanocarriers. So, for example, here you can see different kinds of nanoparticles that have been produced in my lab. I don't know whether it is better this way or this other way. I was told to be this way. They have been produced in my lab, and you see they are different. They look different, their composition is different, but basically, in most cases, what we have is a kind of core material surrounded by a polymer corona or something like a shell, right? And this is because the way we design these nanoparticles is taking into account the properties of the drugs, the drug loading, you know, so the core has to be able to load sufficient, very often we don't talk about sufficient amount of drug. And on the other hand, the coating material helps these nanocarriers overcoming the biological barriers, which on the other hand, depend on the target and on the modality of administration. Obviously, the biological barriers that we need to overcome if we want to deliver a drug to the eye are going to be very different as compared if we want to deliver a drug to the GI tract, or, you know, following intravenous injection with all the systemic barriers and biodistribution barriers. So these are the barriers we take into account. We think about, where do we want to go? Where are the barriers to go there? So I have to put this, I have to select this ingredient, So control release, of course, and many other properties. And well, as I said from the beginning, the drugs we are specialized in are proteins, monoclonal antibodies, polynucleotides. The materials that we normally use are recognized for being acceptable from the regulatory point of view. And what this means for me is that they are already part of marketed products. It's not that I have found they are safe in my lab, it's that they are already on the market. It is true that sometimes we have collaborations with people developing new biomaterials because we really need new biomaterials. Those on the market, are very limited. But in most cases, we try to use biomaterials which are very well known, such as polysaccharides, polypeptides, surfactants, etc. And regarding the techniques, we avoid by all means complex techniques. We try to make the technique, to design the technique as simple as possible, because we have this projection of developing a product eventually, or helping to develop a product. So we know that having a simple methodology which can be scaled up is very critical for achieving that ambitious goal. So, we started around 20 years ago formulating proteins, and at that time it was the age of the PLGA, PEG nanoparticles, and we're actually the first describing the encapsulation of large proteins, such as these antigens, tetanus toxoid, and diphtheria toxoid, a number of antigens which are huge molecules. And we were the first reporting their encapsulation into these kind of PLA, PEG nanoparticles. A few years later, we were also the first reporting the encapsulation of polynucleotides, plasmid DNA, within, again, PLGA and PLA, PEG nanoparticles. And from that time until now, we have encapsulated a significant number of proteins and monoclonal antibodies, polynucleotides, within a great variety of nanoparticles. So this was 20 years ago, and I would like to move towards the new technology we are working on now, which is kind of illustrated here, which seems to be much more complicated than this kind of simple PLGA matrix systems. Now, this one seems to be much more complicated, although at the end of the day, you will see that it's going to be very simple in terms of preparation. So the key here is the selection of the right components of the system. For example, here we have an oily droplet, which is surrounded by a polymer shell conveniently stabilized, and we can put another polymer layer around, and we can put another one, and we can even functionalize this polymer layer with a targeting ligand. Well, we can have such a system, but we can also have, I think this is not very well. Okay, I think this is better. Okay, sorry about that. So we can have the same kind of system, but rather than having an oily core, as it is indicated here, we have a solid matrix, a solid core here. So some interesting features of this new system with which we are working now are those indicated here. Okay, we can modulate the size going from 50 nanometers up to a few hundred nanometers. We can, based on the composition of these coating materials, we can have either a negative surface charge, a positive surface charge, and as I said, we can have a liquid core, a solid core, and we can functionalize the surface. So it's a kind of versatile system which can be used for either large molecules or low molecular weight compounds. So these are the delivery systems that we are using mostly now, and what are the applications? So we are applying these kind of technologies for the development of drugs for treating a number of diseases. For example, we are working on oral delivery of protein drugs and polynucleotides. We are also working on intranasal delivery of antigens, ocular delivery of polynucleotides, and also, of course, on cancer treatments, trying to deliver both monoclonal antibodies and RNA molecules. So I'm going to focus today, we don't have the time to go over all these applications, but I will try to focus on cancer and then to tell you a few words about oral and ocular drug delivery. So regarding cancer, well, we have developed what we call targeted nanotherapies for cancer, and the conceptual mechanism behind these targeted therapies is that we have a dual tissue targeting and also a dual cell targeting. What do I mean by that? When I say dual tissue targeting, what I mean is that we are trying to reach not only the tumor site, but also, and most importantly, we are trying to reach the lymphatics, where the metastatic cells are mainly located. And regarding the cell populations, we are trying to reach the cancer cells, metastatic cells, and I have to say that we are also initiating a project in which we are trying to reach the tumor-associated macrophages. So the benefiting indications, obviously we are trying to deal with cancers which are very difficult, which are incurable, which are very difficult to cure, such as lung cancer or pancreatic cancer, which, you know, would have an important lymphatic metastatic spreading, and they also have a complicated tumor stroma. Okay, so what are the characteristics of these nanocarriers? In this case, this is the kind of system. It's a nanocapsule with an oily core, which is surrounded by a polymer layer which can be functionalized. So basically, the size is small, can be modulated. The materials are regulatory acceptable. The technique is easy to scale up. The product can be freeze-dried. So we take from the beginning in the concept all these properties into account because we want to get there. And something important is the versatility of this nanocarrier in the sense that it can load both small molecules, hydrophobic molecules, but also large molecules. For example, within this nanocarrier, we have associated both small molecules such as docetaxel and also large molecules such as monoclonal antibodies. For example, using docetaxel, we could do a very rigorous biodistribution and pharmacokinetic study. And we found this amazing data. This graph shows the biodistribution profile of docetaxel incorporated within this that we call MPN nanoparticles. You see that following intravenous administration, the drug accumulates massively at the tumor site, which in this case is a longer tumor, whereas the accumulation in other relevant organs is very minor. It is also significant, all these are lymph nodes. So if we take all together, we also see a significant accumulation of this drug in the lymphatics. So as I said before, this delivery carrier is able to target this small molecule, in this case, to the lymphatics and also to the tumor site. But today, I want to concentrate on macromolecules. So we apply the same kind of delivery carrier for the delivery of monoclonal antibodies. And you may have heard a lot about monoclonal antibodies. We use them in nanomedicine for targeting. But today, I'm not going to talk about using monoclonal antibodies for targeting. I'm going to talk about monoclonal antibodies as a therapeutic entity. And as a therapeutic entity whose target is not localized outside the cells, but inside the cell. And I want to make this point because all targeted therapies, all immunotherapies marketed until now are only intended to target extracellular targets, targets that are on the membrane or in the stroma environment, wherever, but never inside the cells. So our challenge has been to design a nanocarrier that enables the intracellular delivery of monoclonal antibodies. So as I say, we have a lot of targeted therapies, hundreds of clinical trials. We are in this fashion wave now of immunotherapy. All of them are intended to target extracellular targets. However, we do have a lot of intracellular targets, a lot of intracellular proteins, which are nowadays considered like undruggable because nobody has been able to reach them. So using nanotechnology, what we have tried is that this monoclonal antibody that otherwise wouldn't be able to go inside the cell because there is not a receptor on the surface, we have tried to put it inside the cell, to deliver it inside the cell, thanks to the use of nanotechnology. So I say this is a paradigm shift, a new concept, because we are trying to target therapeutic antibodies against intracellular oncoproteins. That's the difference as compared to previous work. So what intracellular target have we chosen? Well, we chose one that enabled us to collaborate with some interesting people in the field of intracellular targets in cancer. So our collaborators, Gemma Moreno and co-workers from the Universidad Autónoma de Madrid, they discovered this intracellular oncoprotein, gasdermin B, and they found that it's a very relevant intracellular oncoprotein. It's a new marker for bad prognosis in HER2-positive breast carcinoma. And this gasdermin molecule, when it is overexpressed, and it is overexpressed in many cancers, what it does is to increase cell migration, increases tumorigenesis, and increases the resistance to other classical anti-ER drugs, such as trastuzumab or lapatinib. So it's a very important target. And so they were very interested, this group in Madrid, they were very interested in being able to deliver drugs against this intracellular oncoprotein, monoclonal antibodies. So they produced the monoclonal antibodies for us, and what we did was to associate the monoclonal antibodies to our delivery carrier, and we tested this new composition, the monoclonal antibody with the delivery carrier in cells, first in cell culture, overexpressing gasdermin. And what you see here, these are the cells, the nucleus and the membrane, and what you see, the green spots here corresponds to the monoclonal antibody. So it was clear from the beginning in our experiments that using our nanocarriers, we could deliver this monoclonal antibody inside the cells. And this was very important for them because up until then, they were only able to deliver their monoclonal antibody inside the cells using electroporation. So in this case, they could see that they could avoid using electroporation. And then the next step was to make sure that what was the mechanism of internalization of this monoclonal antibody, and to make sure that it will go out of the endosomes, because we found that indeed these nanoparticles go inside the cell according to a receptor-mediated, non-specific receptor-mediated endocytosis, and therefore part of them, 50% of them were found into the lysosomes, but then we found also a few hours later that the monoclonal antibody could be released out of the endosomes exhibiting an activity. So we checked the activity and we checked the biodistribution as well before going into looking at the efficacy. As you know, one of the critical points when you design a delivery carrier for intravenous administration is the stability in plasma. So we checked the stability in plasma and we found that it was okay. And then we evaluated the biodistribution of these monoclonal antibody-loaded nanocapsules, in this case loaded with the fluorescently labeled monoclonal antibody. And we found here in green again that this monoclonal antibody could reach the tumor following intravenous administration. So this is a proof of the biodistribution. So finally, we went into the efficacy studies. We administered 3 different doses at different times, as indicated here. And then we monitor, as usual, the tumor volume over the time. So we did a number of experiments. In some cases, we treated the animals with IgG, an irrelevant IgG, in 2 cases. In one case, overexpressing gasdermin, another one without overexpressing gasdermin. Obviously, for IgG, we didn't see any response. However, when we administered our nanocapsules with the monoclonal antibody anti-gasdermin, we could see a significant decrease in the tumor growth, which basically indicates that this monoclonal antibody is working in vivo. And this is the first proof of concept of a monoclonal antibody working, being efficacious in vivo, following intravenous administration using a nanocarrier. So regarding the application in cancer, we could say that we have developed this active multi-targeting technology. We can achieve the intracellular delivery of monoclonal antibodies, and we have the proof of concept studies, PK/PD studies. This technology enables us to combine small molecules and large monoclonal antibodies. It's very simple. It's based on self-assembling. You know, like we have the emulsion droplets, and then we add one layer after the other. So it's very easy to produce and very easy to scale up. And we also know that this suspension of nanoparticles can be freeze-dried and is stable during storage. So that was about cancer, but as I told you, we're specialized in designing systems for different kinds of applications. As you can imagine, the kind of barriers we encounter when we administer a protein, a peptide drug, following oral administration are very different as compared to those that I show following intravenous administration. So, like 6 years ago, I decided to coordinate a European project whose aim was to investigate the possibility of using nanotechnology for delivering proteins orally. I know that you may have seen papers talking about that. However, many of the data we have seen in the literature, we found they were not quite reproducible and found very confusing. And so our main goal was to mainly understand how nanomaterials interact with the different barriers of the GI tract, and therefore to see if there is a possibility for these nanocarriers to be used for oral peptide delivery. So this is the composition of the consortium. We just finished this June. After 5 years of working together, 16 groups with different profiles, nanotechnology, chemical biology, toxicology, immunology, a few small companies, and Sanofi as a big pharma. Sanofi was giving us the peptides that they wanted to formulate for oral administration. So you can imagine during 5 years, 5 nanotechnologies working together, we developed more than 1,000 prototypes. And the prototypes were, well, the principle was that they should be based in safe materials and easy technologies that could be scaled up. So we worked with different kinds of systems, nanoparticles, nanocapsules, micelles, and several others, many of them self-assembling systems. And we tested them. We tested these more than 1,000 prototypes for their behavior, for their capacity to overcome biological barriers, and also for their capacity to load peptides. You may, don't believe, I'm not sure you're going to believe that most of the prototypes were discarded because we couldn't achieve the adequate loading. So loading is something that we never talk about, but in fact, in most cases, if you want to think of a potential product for the future, you need to have very high loading. So forget about 1% loading, 0.5% loading. I mean, loading efficiency is important to be as close as possible to 100, but the final loading, which means the milligrams of active ingredient per milligram of carrier, that's a critical parameter that will dictate whether your carrier will have a chance in the future or not. So we decided that we would not accept any nanocarrier with less than 10% of loading. So with this, we discarded most of them. The other important question was to have a capacity to control the release, which was also very difficult. And we discarded a lot of them. But in addition to these drug-related properties, we were testing the nanocarriers with regard to their capacity to overcome, for example, as soon as you administer nanoparticles, almost any kind of nanoparticles, they reach, let's say we avoid the stomach, because the stomach is easy to avoid it with an enteric capsule. But once they reach the duodenum, most of them will aggregate due to the contact with enzymes, pancreatic enzymes, with lipids, bile salts. They will aggregate massively. Let's say that you find a way to minimize this aggregation. Okay, the next step is to deal with the mucus. They have to be mucodiffusive, and the next step is they have to interact with the underlying epithelium. So this is what I'm trying to show here. This is the mucus layer, and this is the epithelium. So assuming that you have been able to make them stable here, they have to cross this mucus layer, and then they have to interact with this. So we wanted to know if these nanocarriers were able to stay here, if we were able to cross or stay here, or even to be translocated. And we found that indeed, depending on their size and their composition, some of them remain here, stuck in the mucus, others were able to reach that level, and others were able to be translocated. For example, in one specific prototype developed in my lab, we found a massive accumulation of nanoparticles at the epithelium level. And this is what you can see here in green. This is a cross-section of a human intestine And these are the villi and the cells with the nucleus. And you see all this kind of film is because the nanoparticles interacted with the epithelium and remain stuck there. So at the beginning, we were kind of frustrated. We said, okay, these are not going to work for delivering insulin, for example. But then we realized that this kind of system might be very useful for the treatment of local diseases. You know, for example, Crohn's diseases or intestinal bowel diseases. So we think this as a candidate for that kind of purpose. But we also developed other nanoparticles which were able to deliver, for example, insulin across the intestinal barrier. And some key properties of this type of nanoparticles that we call enveloped protein metal nanocomplexes. This is a photograph. They are kind of square, very small, and they have these properties: a small size, neutral surface charge to avoid the interaction with the mucus and with the enzymes, stable in intestinal fluids, high protein loading, more than 10%, prevention of degradation, and prolonged protein release. And just for you to see some of the in vivo data, we are showing here the decrease in the glucose levels following oral administration to rats. This is the control, and this is our formulation. So we found that indeed this formulation led to a significant enhancement of the effect of insulin following oral administration. This formulation is now being tested in pigs because it was the most promising coming out from the consortium. Most of them didn't work in terms of facilitating the delivery of insulin, but some of them could work for other peptides and for local purposes. So the conclusions for this specific application, oral peptide delivery, is that we could tune the interaction of nanocarriers with intestinal mucosa. So some of them stay at the mucosa level, others were able to transport insulin across. We have in vivo proof of concept studies, in particular PD studies. We see, using this system, the possibility to deliver proteins and RNA molecules, and again, easy to scale up, and the possibility to have it as a freeze-dried powder. We always mention that because we think they are essential properties. And finally, just a couple of words about our work in the field of ocular drug delivery. You know, very different barriers. I mean, the ocular surface, one of the problems is that the amount of drug you can administer is very low. And also, the cornea is very well organized. It's a very difficult barrier to overcome. So a number of years ago, 10 years ago, as you can see in this paper, we wanted to try to see the possibility of using nanoparticles for delivering polynucleotides to the eye. And this idea was based in previous evidence of the capacity of some nanoparticles to overcome the corneal barrier and to go into the cell of the corneal epithelium. So what you see here is a transfected cornea. So basically what we did was to associate plasmid DNA encoding the green fluorescent protein to hyaluronic nanoparticles. We administered these nanoparticles as a drop to the eye of the rabbit. Then we removed the cornea and we looked at it by confocal microscopy. We could see how the eyes of the rabbits became green after 1 day, and the duration of this expression was for more than a week. So based on these experiments from 10 years ago, we have gone further. These nanoparticles were made of chitosan hyaluronic acid, and we wanted to change for more friendly biomaterials. And nowadays, we have a couple of prototypes that we hope are going to be promising for RNA delivery to the eye. For example, in this case, we have protamine nanocapsules in which we have an oily cord surrounded by a protamine layer. We can make them from 200 nanometers down to 80 nanometers. So we associate RNA molecules to the surface, as it is indicated here, and then we administer it to the eye of the rabbits. We found that we can produce these nanoparticles with either negative, positive, or neutral charge. They are stable in the lacrimal fluid. That's a requirement, stability in the lacrimal fluid. And then they have a very high RNA loading. I think we are in a kind of rush. This is another prototype that we developed for RNA delivery to the eye. In this case, we have a nanocomplex of RNA and a penetration enhancer conveniently stabilized by polyglutamic acid polyethylene glycol. So in terms of ocular drug delivery, we are now working in collaboration with Silentis company and in one European consortium and a couple of consortia within Spain. And so far we can say that we have reached all these properties for these nanocarriers. Very high RNA concentration. This is very difficult to reach. Control RNA delivery. The systems are biocompatible and biodegradable. We have seen that using rabbits. The system is stable during storage. That's a key property for ocular treatments. And we have found in preliminary studies an enhanced corneal penetration. So we don't have more time. So just to conclude about the rational design, it is certainly true that the understanding we have about our biology is still limited. So we really need to go for a deeper understanding of the biological barriers in order to make significant progress in our field. We also need more biomaterials, and we need to know them well. We need a rigorous knowledge about the biomaterials and their chemical functionalization. And of course, we need also the adequate technology design in the sense of the formulation work, adapting the delivery carrier for specific protein molecules or polynucleotides, and what I mentioned before, the necessity to scale up the technique. And I want to end by mentioning that the analytical methodologies, especially when you work with biomolecules, with proteins, monoclonal antibodies, are also critical in terms of letting us to advance in this field. We spend quite a lot of time with analytical hurdles. So just to thank my research group in Santiago de Compostela. Our lab is here at the top of this tower. And since we are short of time, I want to thank my collaborators from the USC, also the external collaborators. And with the permission of Victor, I will make a short publicity because I don't know how many people here know about the Control Release Society, but I just became the president-elect of the Control Release Society. And so for those of you who don't know the Control Release Society, this is, I would say, pioneering society highlighting the value of drug delivery technologies since the 70s. And the meeting this year is going to be in New York. So a very attractive place, and hopefully the following year is going to be in Europe, hopefully in Spain. We will see during my presidency. And in addition to this, the Control Release Society has what we call local chapters, because the meetings of the Control Release Society are like 1,500 people, so they are very good for networking. But sometimes we want to go to smaller meetings, and this is the case of the meeting of the local chapter in Coimbra, Spanish-Portuguese local chapter of the Control Release Society, which is going to be held in Coimbra. Thank you very much for your attention. I'll be happy to answer any questions you have.
Thank you.
Thank you.
Thank you for the presentation. We open the session for discussion. Any question here?
Good morning. Great talk. My name is Stefan Wachikowski from University of Duisburg-Essen in Germany. I was very impressed by your intracellular targeting. And I learned that you use antibodies and load RNA maybe by physisorption. But for medical application, as you know, sterilization is very important to make your emulsion or whatever, your drug, sterile. Can you give some recommendations how to consider this while synthesizing your components, that at the end everything is going to be sterile?
Yeah, that's a very practical point. I have to say that we try to be very practical in our approaches, but not that far, as you mentioned. But it is true that we think that most probably, we never try to sterilize the final product as such. So what we think is maybe the aseptic production, as it is the case for other nanocarriers, would be the best way to sterilize the different materials and then to mix them together in sterile conditions, which is going to be relatively simple, because the technique does not involve, high energy sources or organic solvents. It's just based on mixing. So mixing sterile solutions in an sterile environment, I think, is going to be the easiest way to have an sterile product.
So you would not recommend, you would recommend to avoid having any contaminants rather than post-sterilization of your formulation?
I see this as an easy way, but obviously the first thing to try is if the system will survive under sterilization at the end, right?
Probably not.
Okay, thank you.
Neil Thomas, University of Nottingham. With the intracellular delivery of the antibodies, does the constant region actually have any role at that point? Obviously, you could use Fabs or single-chain FVs to actually bind to your targets. So why would you want to use a whole antibody at that point?
Okay, I know that there are other people producing nanobodies trying to reach the same goal. But what I'm presenting here is that using the classical monoclonal antibodies, easy to produce, no chemistry around, even in that case, we are able to deliver them. So obviously, the technology could be applied for other Fabs or nanobodies or whatever kind of molecules. But I think for us, the aim here was to show that we are able to deliver a monoclonal antibody without any further manipulation, just as it is produced. So we think this is of value. But obviously, working with the small molecules would also...
they would help give you a higher loading, potentially, because obviously you're using things that are half the size of the whole antibody.
And we envisage that for the future, this is true. However, in general, monoclonal antibodies are very active. So in that case, for the oral project, the loading was very high. In that case, for the monoclonal antibody, it was not necessarily a high loading. So for the in vivo experiments in animals, of course, but thinking of the future, you are right, there are other options.
So we thank the speaker again, and I think we are ready for coffee. Coffee room is in the